Hi, thanks for viewing this talk. I've been fascinated by monkey flowers for about 40 years, and the group I'm going to talk about has never really been the most interesting to me, but that all has changed with the new treatment in the Jepson manual, Guy Neesom's work on the monograph for this group, and recently 4 new species have been discovered and described. So those are new discoveries I'd like to share with you. And right off the bat, in a preamble, I'll start by giving the answer to this. The talk started out, what is Erythranthe guttata exactly? And I think for most people, what Erythranthe guttata is any yellow monkey flower growing in a wet seepy area that looks like yellow common monkey flowers, and without realizing that there's now many options, different species that those could be, are happy to call everything Mimulus guttatus. It is, I think, the actual Erythranthe guttata is very wide ranging in California, but if you look at all these dots here, I think it probably represents many different taxa. So what I'm going to cover in this talk is about this section that Erythranthe guttata occurs in. I'm calling it messy diversity. It's not all worked out. And the new morphology-based taxonomy that's in the Jepson flora now, there have been 4 new species discovered and described very recently. And then on a side note, and perhaps leading to dovetail with this morphological work, there are now hundreds of scientists working on plants in this group, but in a more basic science mode to learn about how genetics works, etc. And then I will finish with a ultra shortcut to how we're going to actually identify these things in the field. So again, what are we talking about? We're talking about a group of Erythranthe, which when the old family Mimulus was, or the old genus Mimulus was split up, Erythranthe was one of the major new genera, and this grouping Semiola is one of the groups within that includes the common yellow monkey flower. And what morphologically gets them to key out in this group is the characters of the calyx, with the upper calyx lobe pointing outward as a finger-like projection, and the 2 lower calyx lobes bending upwards. This is all in fruiting and maturity, and that's other species of Erythranthe do not have their calyx lobes behaving this way. There's also almost always a platform on the lowest corolla lip that kind of bulges upwards, and that doesn't always close the corolla mouth. Pollination can occur in some species, but here's just some pictures of what is Semiola, this subgenus, and what is not. And just because it's a little yellow Erythranthe doesn't immediately put it in Semiola. The new write-up in the Jepson Herbarium was completed in 2018 by Naomi Fraga. She gave a great talk here either last year or the year before, talking about all that work and breaking up Mimulus into smaller genera. But I really recommend you look at this if you want to, you have to look at this if you're going to try to put species names on the Semiola. Her treatment, though, did rely on this monograph that Guy Neesom, a amazing taxonomist in his 70s now, who got a contract from Flora, North America, to work on this group. And as of the monograph in 2012, there were 31 species in North America, and he's continued to publish new species. And I think it's up around 40 species for the whole area. And with respect to California, here you can just see how over time, the number of recognized species in the publications that we're using to try to identify plants has shifted around a bit. The eFlora has 17 species in it for California, and then there's been 4 since then. And let's take a look at those. David Keel, a retired professor down in San Luis Obispo, described this species practically from his backyard or practically in the city of San Luis Obispo on serpentine, as the name indicates. And it has some morphological distinctness, and it occurs very closely to populations of Simiola monkey flowers that are not on serpentine, and they are pretty morphologically distinct. So he described that. Guy Neeson looked at some plants that Anne Howell had discovered in her botany work in the Sierras and Mono County. And this plant was described and named after Anne, and it's still a very localized population on Highway 108 near Sonora Pass. It bears some similarities to some other very restricted monkey flowers. I've not seen it. I'm looking forward to checking it out. And then if you know Matt Berger, everything he does always makes you say, wow. And yeah, apparently he discovered not 1, but 2 new species of monkey flowers, kind of very close to each other, not very similar at all. So really kind of amazing set of finds. And Guy Neeson named this one after Matt. What makes it unique is the lobes of the bracts are connate, they're fused, and there's only one other species, Glaucessans, in this group, Simiola, that has those fused bracts, and it occurs way up in Butte County. So this was very unique morphologically and had some other interesting characters. So that was one new one, and only a few miles outside in the Big Pine area of Owens Valley, and then just a bit down the road near Lone Pine, Matt discovered this other monkey flower, and it's pretty amazing to me. I mean, it looks like the Simiola group in many ways, but what really stands out are there's these structures where the nodes of the stems come together, and sort of bulbous structures, and they're exuding a liquid that seems to be attracting and feeding ants. So there's probably some story there that hasn't been figured out completely yet. And some other unique things about the plant are the way it sort of is very close to the rock face and kind of hugs it, which there's a number of different mimulus that occur on cliffs and have that same behavior. So really neat stuff. And so there's still a lot to be discovered. And again, yellow monkey flowers, they're not all the same thing. They're not this thing called Erythranthe guttata. There is an amazing diversity. Shifting gears a little bit, but I really wanted to talk about this, if possibly a bit of a coincidence, but in the past 25 years, a number of these species in Simiola, and they're often thought of as still Mimulus guttatus and I guess friends, but there's a few thousand papers that have been published, 100 or more researchers, and they've really found it to be a convenient plant to work on with really short turnaround time, easy to grow, a lot of variability. And it's sort of joined the cadre of the fruit fly, lab rat, And then another plant, Arabidopsis thaliana, has had thousands of papers published on it. It's another model organism. So now we have a North American model organism drawn from the Semiola section. And here, this just, Dina Grossenbacher shared this with me, that showing in Web of Science, just the explosion of papers being published using these plants. A point I'll make about it is it's all very great scientific work and everything, but I'd say so far it really hasn't led to clarification of taxonomy and sort of the classification of how many distinct entities are out there on the California landscape and what numbers, what names we should give them and how they're related, and certainly not, sort of making it crystal clear. Now, the molecular and genetic work sometimes gives a very clear picture on how messy and sort of not clean situations are. So I think it'd be super exciting if these people with their high-powered genetic tools and really amazing understanding of what the plants in this Semiola section, how their genome is structured and everything, but there really needs to be studies that pick very morphologically distinct populations and then run them through the genetics and get cladograms, which really can shine a brighter light on what we're seeing. And I guess I hope that some student researchers or groups of students will come along and do some of these bridging studies. Looks like I've left myself a few minutes for what I think might be sort of the take-home information from this talk. And so now we have 21 different taxa in California that many of us would have previously been inclined to glance at and go, I don't know, looks like common yellow monkey flower, and either thinking, well, those are really hard to key out, or I haven't even looked at the new key or anything. And so here's a strategy, and it's a sort of flawed strategy, but 14 of these 21 species are pretty localized and restricted to very small parts of California. And so this is showing 17 of the species that this is where they've been described and identified. And so it's sort of cheater botany. You're never gonna make range extensions or that sort of thing. But as you go through the keys, you can do a lot of exclusion of things by not being in the geographical area where they are known to occur. So again, maybe if I was a real botanist, I wouldn't be saying these sorts of things, but that's how I usually go through keys, is when I can't figure out couplets, looking for geography as well. And that leaves us with what appear to be from Guy Neeson's monograph, 7 more widespread taxa that his morphological analysis have led him to the conclusion that these are taxonomically distinct. And I'm still trying to find these things in the field and key them out. I'll admit right off the bat that I haven't seen what I've identified as Corallina in the first group or Arvensis in the lower group, and hope to keep just getting out there and looking at these monkey flowers. But otherwise, these are the species in yellow, guttata, microphylla, and nasuta, I think are what you're generally gonna encounter as widespread monkey flowers in most field sites. And so the real distinctions now are perennial versus annual. And if you want to call something Erythranthe guttata, it needs to be a perennial with well-developed rhizomes or stolons, and not a little, or tending to be a smaller fibrous rooted or tap rooted plant. So right off the bat, that's restricting what is guttata. And I think both microphylla and Nasuta are very widespread throughout California. They tend to occur in areas that start out wet in the winter and spring and then dry up. They're annuals. The big difference between microphylla and Nasuta is microphylla is outcrossing, the corolla is much larger and is being pollinated. Nasuta tends to have very small flowers. Sometimes they don't really open and can be very tiny plants. And with that, I believe I am done with my 15 minutes. So thank you for joining me for this talk. And I hope there are some questions.